All right. Hello, everybody. Thanks for joining us today. This live stream is a little bit different from the ones that we have been doing. This is all for the people educating. Although if you're a high school classroom here too, we're happy to have you here too. So today we are joined by my buddy Rob O'Malley. Welcome, Rob.
Hello, good to
Thanks
be
for
here.
coming. Yeah, so today we're going to
Yes.
be talking about how we can communicate personal genetics in classrooms and out in communities in informal science education settings. I'm going to hand it over to Rob pretty much right now. Just quick housekeeping. If you have any questions as we're going through the live stream, feel free to put them into the Q&A at the bottom. I will read them out loud when we get to the Q&A portion after Rob goes through his slides. Rob, I'm going to hand it over to you.
Okay, great. Thank you so much, Sarah. It's really exciting to be back here. I've been a fan and supporter of Skype-A-Scientist really since it started. So it's always exciting to kind of re-engage with this community. You mentioned to Sarah, I just got my Squid Facts, Eel Facts calendar this year. So you can check that out on the Skype-A-Scientist website. So I am Dr. Rob O'Malley. I am here from Personal Genetics Engagement and Dialogue, or PGED. And today we're going to be discussing personal genetics engagement in classrooms and communities. This presentation was developed with educators and science communicators in mind, but everyone is welcome to attend. I know there's a high school on and maybe some other schools. You'll be very welcome. I hope this will be interesting. And I've tried to make this really accessible and engaging. So we're talking about engagement, and I tend to believe that a great way to learn about a thing is to do the thing and not just talk about the thing. So we will be talking about the thing of engagement, about genetics, but we'll also be doing the thing together. So there'll be some engagement points for us to think about some personal genetic topics together, which is intended to really model the kind of prompts and framings that could be done in classrooms or in engagement contexts. And I have tried to leave time at the end to talk about for more free, open conversation about, what this work looks like in practice. I do want to note at the beginning, though, that everything that I'm going to be talking about today, all the resources that my program has developed, are all freely available on our website, pged.org, and I'll post that at the end as well. Okay. All right. So, as a first point, I want you to invite you to share in chat if you're comfortable. What is the first thing you think of when you hear the word genetics? So, what comes to mind when you hear genetics? Short answers are good here. I'll pause and let people ponder that for a minute. Okay. Excuse me. DNA and inheritance, sure. Inheritance. C plus. I also got a C in genetics in college. Their thoughts? Diagrams? You can put more than one answer if you like. Some people's mind jump to Punnett squares. See a lot of Punnett squares in genetics. Or things in the news, perhaps. Traits, sure. Disease disorders. Okay, so that's a good one. So thinking about where it intersects with human health. Just a few more seconds. Any final thoughts? Inscrutability. Yeah, it's a little mysterious, as with many things in biology. Um, Okay, great. So, a range of responses there. In keeping with, I think, DNA that makes us what we are, that's a great point. That's actually segues into something I'm going to talk about later. Um, so, genetics is, of course, a science, but when thinking about in public spaces and discourse in classrooms about genetics in our lives or genetics in the news, things like that, it can provoke a pretty wide range of reactions, and that range of reactions is part of what we're going to talk about today. The framing for our time together is I'm first going to talk a little bit about how our program frames the concept of sort of personal genetics, and why we take that approach, and how we approach the challenges of engagement in classrooms and communities around this topic. We're going to briefly explore a couple of examples of where genetics advances are intersecting with people's lives, and then hopefully leave some time for more open-ended Q&A. And again, there's going to be some interaction points throughout because I believe strongly in doing the thing rather than just talking about the thing. So our program, PGED, is based at Harvard Medical School. For 19 years, we've worked to raise awareness and inspire curiosity, reflection, and dialogue about genetics, with the goal that we're helping to envision and create a future where genetics serves all through widespread awareness, active engagement, and individual empowerment. Speaking for myself and who I am, so I am a white cisgender and heterosexual man from a middle-class background and a U.S. citizen. I speak English and some Spanish and Swahili. I am a parent and I am agnostic, though I was raised Catholic. And my academic background is in biological anthropology, specifically primate behavior and ecology. And I've been working in public engagement with science for over 9 years, first at the American Association for the Advancement of Science, and now at PGED, where I've been for about 3 years. And I am not a geneticist. I mentioned earlier I got a C in genetics in college. But I have a lot of experience in facilitating constructive conversations about science across differences of perspective and worldview, including on potentially contentious topics like human origins and evolution, and other elements of human biology and anthropology. And beyond my work at PGED, I'm also the Education Committee co-chair for the American Association of Biological Anthropologists, or AABA. And I'm joining you from Rockville, Maryland, which is where I live. And please feel free to introduce yourself in chat as well, if you're comfortable doing so, just who you are and where you're joining from. So just very briefly, this is just an orientation, just in case anybody appreciates this kind of grounding. So when we're talking about DNA, that's really at the heart of the field of genetics. This is a code consisting of four letters, A, G, C, and T. And this deoxyribonucleic acid, that's what DNA stands for, is found in nearly every cell in our bodies. And nearly every cell in our bodies contains this code, this DNA code, that is about 3 billion letters long. And the complete copy of this DNA code is called a genome. And this DNA code is, there's lots of ways to think about it in terms of metaphor, but one way to think of it is it's kind of a cookbook full of recipes that interact with the environment to create you and me and every other living being on the planet. And so genetics encompasses everything from the study of DNA structure and function to heredity to patterns in living organisms at every scale, including how they interact with the environment. And we're going to be talking quite a bit about how powerful DNA can be for helping people learn about themselves, for sharing information with relatives, for addressing societal challenges. But it's important to recognize that DNA alone does not define us or any other living thing. This is a common thread that unites all of life, that unites all people. But the genetics of living organisms, including people, often interact in complex ways with environment and behavior to impact all elements of life, from growth to development to ongoing functioning. And there's a great deal of flexibility or plasticity in living systems that isn't defined by the code of our DNA. But it is very important. And at PGED, we frame personal genetics as an exploration of how genetics impacts people, families, and communities. In addition to the social impacts of genetics, personal genetics can also highlight benefits and concerns related to the use of genetic technologies, ranging from individual health to medical research and other broader societal enterprises to the living world. And like many fields of science, genetics is advancing quickly. What we have here are two types of DNA sequencers. One that was used in the Human Genome Project that took 13 years and $3 billion to sequence the very first human genomes. These were enormous machines that took a long time to run. But by 2025, um, a genetic sequencer can cost as little as $600 and can be done in less than 24 hours. And the machine that does that is about the size of a TV remote. And so the cost and time for these analyses continues to decrease and become more efficient every day. In the context of that rapid advance, it's helpful to note that the US public's familiarity with basic concepts in genetics, genetic terms, and elements of the field have shown a lot of improvement over the last decade, according to a 2022 study. So people know more about genetics than they did 10 years ago. Public attitudes about genetic technologies related to the detection, prevention, and treatment of disease, which is one growing and important element of genetics research, are viewed very favorably. At the same time, the public tends to hold negative attitudes towards technologies related to human enhancement or selection of traits. So, for example, modification of people to be more athletic or more intelligent, things like that, which are largely hypothetical, but potential applications of genetics. There are concerns in particular about privacy and discrimination, such as in the context of insurance coverage or employment. So people may be misusing your genetic information to decide who gets hired and what kind of health insurance you're allowed to have. And about half of US adults express concern that the pace of science and technology advances, including in genetics, is making life change too fast. People's feelings about other genetic applications beyond human health are often ambivalent. For example, using gene editing to control mosquito populations or enhance nutritional quality or drought resistance of food crops. We can and should also consider genetics in a broader context. Distrust of science and of scientists and of medical professionals is in many ways a historically informed perspective. There are no shortage of examples of scientific ideas, including from the field of genetics, that have done tremendous harm. For example, the infamous Supreme Court case, Buck v. Bell, in 1927, affirmed the right of the state to forcibly sterilize people it deemed unsuitable for reproduction, a finding that has never been explicitly overturned. This decision took place in a period where eugenics, the idea that humanity can be improved through breeding better people, was a completely mainstream and widely held idea, one embraced by prominent scientists, major scientific societies, organizations, and institutions around the world, including my own university, Harvard. And there's a tendency to view this history as only history, to treat it as a regrettable chapter that science has moved beyond. However, science, including the field of genetics and related fields, has in some cases continued to be a source of harm in practice and application, particularly for often marginalized and minoritized communities. As noted by Dr. Crystal Sosi, a Dene geneticist, and her colleagues, speaking on indigenous relations with biomedical research, she notes that harms from biomedical research for indigenous people are ongoing, not just in the past, within a research framework that is too often done to and about indigenous people rather than for, with, or by them. So many of our resources in our program frame personal genetics around ethical, legal, and social dimensions of the field. We do this because the field is ever evolving. We want to be supporting robust public and policy discourse about these changes. We want to recognize the very real issues of inequity that continue to persist in science and medicine and its applications. And also recognize, as I just referenced, the history of harms, and in some cases ongoing harms, from science and genetics in particular. And by recognizing that social, historical, cultural context, we can hopefully mediate or avoid repeating some of those harms. And in thinking about personal genetics in classrooms and in other learning contexts, we found that this approach often provides inroads into science for people who feel that science isn't for them or a place they feel comfortable. And so that's an important element of why we advocate for pulling some of this into classroom education. So in our work, and this is pretty broad strokes, and I'll hopefully talk about it a bit more later. In our work, we try to avoid advocacy for science, so we're not rah-rah science or any particular technology. We don't encourage people to think positively or negatively about genetics. We try to avoid pushing for particular outcomes. We do strive to engage in sharing and learning about science, encourage reflection on perspectives, particularly perspectives that people may be less familiar with or that they disagree with. Use timely and relevant information, so be up to date. And in general, just kind of engage with science in a cultural, historical context. In practice, this means lots of sharing and learning, centering values, identity, and worldview as part of science practice, and really trying to build stronger relations between science and broader societal concerns. Um, and this can be hard sometimes, but this is what we strive for. So the kinds of communities and partners that our programs work with include classroom educators, K-12 educators, but also university instructors and scientists who do engagement work. So that's been a longstanding interest of our program. We've also worked extensively with faith communities. Most people on Earth claim a faith identity, as do many scientists. And so faith perspectives as part of worldview and values informs how people engage with science. So we recognize that as important. Recently, we had a film screening and discussion of science and faith at the Boston Museum of Science. We've co-organized the Genetics and Spirituality Conference in Omaha, Nebraska for faith communities. And this year we had a session on science engagement with faith communities at the 2025 American Association for the Advancement of Science annual meeting. So this is very much something that is part of our work and part of mainstream science engagement. We've worked with storyteller communities to incorporate genetics into film and television, both fiction and documentary work. Most recently with the Social Impact Entertainment Society to develop resources and programming about gene editing and so-called de-extinction efforts, where people are creating things that are maybe analogous to extinct mammoths or dire wolves or things like that. And we've worked extensively in informal science learning spaces, from after-school programs, events at museums and libraries, and local community events, including this year some science fiction and pop culture conventions. And it was a real treat this year to learn about gene editing and talk about gene editing with some of the X-Men, as you can see down in that lower corner, because they would know, right? And historically, we've also hosted events for the Congress of the United States on gene editing, genetics and space exploration, some other topics. But even in this context, we're not pushing for specific policies, which is somewhat unusual in the context of congressional briefings. We're advocating for informed decision making and trying to create, again, those opportunities for sharing and learning about sometimes difficult and challenging topics. And more information about all of this is on our website. And resources developed through these partnerships and with these audiences in mind for classrooms, for scientists, and to support engagement with genetic topics across broad segments of society. All of this is freely available on our website, and I'm happy to talk more about that at the end. But these are just some examples. We have games, we have discussion guides, we have full lesson plans with keys and quizzes and things like that for people who want to go that route. So, having provided that background, I'm going to spend some time briefly looking at how we approach some personal genetics topics in the classroom or in other contexts. And all of these are topics that we explore much more deeply in our lesson plans and other resources. So let's start with genetics and health. So, um, so uh, uh, Sarah's going to post a poll and invite you to respond and share your thoughts. And the question is, I would want to find out my likelihood of developing a life-limiting genetic condition, so some serious genetic condition, only if there are ways to prevent or treat it. And I'll give you guys a moment to respond to that. So you would want to find out if you have a serious genetic condition only if there's something you can do about it, is another way to think about it. And um, if you have a chance to respond to that, then if you're comfortable doing so, just post in chat what kind of informed your thinking, very briefly, if you can. And if you're not comfortable doing so, that's fine too. So what informs your thinking there? Why did you pick what you did? Okay, so one respondent notes, want to make life plans accordingly. Okay, so maybe it's helpful to have that information if you know that like maybe you have a high risk of developing Alzheimer's or Huntington's disease or some other very serious condition. Ways to avoid future complications, sure. So even if there's no way to maybe effectively treat it, maybe there's some mediation that could occur. And someone who said, I want to know no matter what, because just to kind of know what's coming down the pike. Absolutely. Okay, so this is the type of thing that can really get people talking, and even like in a classroom setting, this is the kind of activity that can get people to start sharing and learning what they know and why they feel that way. So to kind of then move into some background on this, and I did that deliberately to have the poll and then talk a little bit more specifics. So health conditions could have many different causes. Viruses or bacterial or fungal infections can cause people to be unwell for a short time, but a person's DNA can also affect their health. Different variants in DNA have already been known to, for example, provide some protection against certain types of infection. For example, norovirus infection, we know that there's a genetic variant that provides protection for that. There's other genes or genetic conditions that are very strongly linked to cystic fibrosis or Huntington's disease or other and more recently perhaps Alzheimer's. So we know that genetics can play a significant role in shaping a person's health, and developing certain issues. And so it is possible in some cases to identify those differences. So reveal whether an illness is genetic, if other tests have failed, identify future health risks, provide information about how to respond to certain medications that can be important in treatment plans, and also be thinking about uh, future generations. Oh, so do I need to share the results? There we go. I didn't realize I needed to do that. So yeah, so we had a range of responses, are pretty spread out there, which is not atypical when we do these types of polling. So you all can see that? Yes, Sarah, they can see that, the responses. Yes, okay. Um, okay, so right. So some genetic tests, so genetic tests are not all the same. Some require the participation of a medical professional. So these are clinical tests that you might have ordered by your doctor. These are not the same as direct-to-consumer tests that you could buy at CVS or online. So clinical tests typically require medical referral. They involve medical professionals. They may be quite expensive, um, and typically can either require a blood sample or a cheek swab. In contrast, direct-to-consumer tests can be purchased without a doctor's request or participation or knowledge. So these are, direct-to-consumer DTC tests from, like, for example, Ancestry.com or 23andMe, that type of company. These are available for purchase, usually do not come with the support of genetics professionals, can be much cheaper, typically use a spit sample or a cheek swab, but they're after the same thing. They're after, coding your DNA. They're often also analytically limited. They are not always looking at the complete range of conditions or the complete genome that a clinical test might do. So, for example, a clinical test doing a cancer screening might analyze thousands of variants, whereas a DTC testing company that's looking at cancer risk might only look at 3 or 4 variants. So, very different levels of resolution and uh, uh, intensity. And since this testing is first emerged in the early 2000s, millions of people have sent their DNA samples for testing, seeking information about their traits, their ancestry, and health. And while that can be an exciting way to learn about yourself and your body, there are concerns that providing people with genetic information without medical professional support can lead to people misinterpreting results, feeling false senses of security or concern, or make medical decisions based on very limited or incomplete information. And as previously noted, it's like these tests have different levels of resolution. One test, a very expensive test, might be analyzing the whole genome. Other tests might just be looking at particular segments of the code or even individual base pair differences. So looking at like even a single A or G in a particular place. Genetic tests can be used to identify potential risk factors. So there is a diagnosis element. People with rare conditions may learn about those conditions when other approaches have failed, and that can play an important role in disease management and prevention. It can also be used in a predictive way to predict a person's risk for developing a condition in the future. As one real life example, you may know that Angelina Jolie in 2013 had chose to undergo a double mastectomy, which is a surgery in which breast tissue is removed. And she did this because a genetic test had identified a variant in the BRCA1 gene that greatly increased her risk of breast and ovarian cancer. She later revealed that she had her ovaries removed for the same reason. And some people believe, now obviously this was a personal medical decision, some believe that preemptive double mastectomy was a drastic and unnecessary choice since she didn't yet have cancer. But her argument and her reasoning, on which others, weighed in on in support, was that by undergoing this surgery preemptively, she greatly reduced her chances. And that was, informed by robust medical testing. Many people have had the experience of taking a medication that works well for other people, but not for themselves. And one potential explanation for that is genetic differences among people. A category of testing called pharmacogenetic tests are used to help healthcare providers identify the most effective medications or dosages for a person based on their DNA. And, because a range of reaction for a particular medication might have no effect, it might have some adverse effect, and it could even be fatal. So getting to the bottom of that can be really important. A real life example of this is examining genetic reactions to codeine. So this is an opioid that's frequently prescribed for pain. For the drug to be effective, the body must be able to convert codeine to morphine. And this is done with the help of an enzyme called CYP2D6. And that enzyme is linked to a particular gene in the genome that has different variants. And different variants of this gene significantly impact how quickly an individual can metabolize this drug. And if the dosage is not calibrated correctly, it can have very serious consequences. Some people can be characterized as typical metabolizers, the most common metabolizers, experience effects from a typical dose of the medication. So their body converts codeine to morphine at kind of what's considered the typical rate. However, there are people who have versions of CYP2D6 that make them slow metabolizers that convert codeine to morphine at a slower rate, meaning that they will get a very limited effect from the same dose relative to a typical metabolizer. And then there are people who are rapid metabolizers who convert the same dose of codeine into morphine very, very quickly, which could potentially cause a life-threatening spike of morphine in the person's body, because again, this is a powerful opioid. So what your genes are, the particular variant for this particular gene, has a big impact on how you will respond to this fairly widely used pain medication. So as a result, as this has become known, the FDA has approved genetic tests for determining what level of metabolizing rate a person has. However, these tests are not routinely used in clinical settings, so they are available, but may not get used, which is maybe a broader conversation about equity and medical practice. Individuals who have a personal or family history of not responding to medications as expected will often be encouraged to seek genetic analysis for an explanation, especially before they're prescribed codeine, but it's not necessarily standardized. In 2017, the FDA also restricted the use of codeine as a treatment for children and recommended that breastfeeding women avoid taking codeine to prevent adverse effects on nursing babies. Another application for genetic testing is for pregnancy and reproductive health. Genetic testing can help people conceive if they're having trouble conceiving. And I can share that there are people in my family who have done this to kind of suss out why they're struggling to conceive and have children. To learn more about the health of pregnancy during pregnancy, guide embryo selection, and also to have some information about potential treatment plans in utero, because that is possible, or after birth for infants who may have particular genetic conditions. And then much more recently, and this is where a lot of excitement is in genetics applications, um, it's researchers are actively exploring treatments to address genetically based conditions at their source. And gene or genome editing is one form of gene therapy that is modifying genetic code of individuals. Genome editing technologies allow scientists to target particular segments of a person's DNA and make changes that impact that function. And if a known genetic change causes a person's health condition, this approach might offer an effective treatment or at least prevent a condition from worsening or continuing. And one, again, real life example of this that's been in the news recently because it's been very successful is gene editing for sickle cell disease. You may have heard of sickle cell disease as one of the most common blood conditions in the world, affecting millions of people worldwide. And sickle cell disease is caused by a single change, a single change in a person's DNA in one particular gene. So it's a very simple genetic system, making it a good candidate for gene therapy because we know exactly what part of the genome to target. And in 2019, Victoria Gray became the first person to undergo a clinical trial using genome editing to modify genes in her bone marrow cells, which in turn produce red blood cells, effectively curing her of sickle cell disease. And this treatment ended a lifetime of sudden pain attacks and debilitating fatigue. And first successful genome editing based treatments for sickle cell disease and related blood disorders were launched in 2023. So now many people have undergone this treatment. While the success of these treatments is very exciting for scientists, patients, and families, and providing a lot of hope for other potential conditions, there are real concerns about long-term impacts on individuals because gene editing remains a very new technology, and also very real concerns about equity. The cost of this treatment currently is about $2 million for a person. Um, and given that millions of people worldwide suffer with sickle cell disease, experience life-limiting impacts due to sickle cell disease, how quickly will this therapy reach people around the world who need it? So now we're going to shift to, I know it was a bit of a whirlwind tour, but now we're going to shift to another area of personal genetics that's of great interest, which is ancestry and identity. And Sarah might have the next question queued up, but if not, we can just read it. Okay. So for this question, same kind of structure, agree or disagree with this statement. I would use a commercial ancestry test. Sorry, let me advance one more here. I would use a commercial ancestry test to find and connect with unknown relatives. So this is using some of the same commercial tests that are used to assess, in potential health information. Now we're looking at ancestry. What parts of the world you came from? Is there a percentage of your DNA that they can ascribe to Neanderthals? Things like that. But very often this can lead to connecting with unknown relatives. Okay, and again, as before, if you're comfortable doing so, feel free to share why you would agree or disagree with this statement. I'll give people a minute to ponder that. Might be fun, learn about second cousins you didn't know about, who you're related to, sure. Maybe discover some famous person in the system. Okay, so connect with maybe so interestingly. So I like this comment. Connect with traditions and recipes that were not handed down to me. So reconnecting perhaps with um, a heritage that you were unaware of or that you feel some distance to. Um, Maybe an ancestry test can provide a deeper connection, and then through family, through relatives, that can be part of that. Okay, so yeah, so again, like a range of responses here. Okay, and someone notes, yeah, this is actually leading into our next thing. Someone has a comment, would not necessarily want my personal information to be used for commercial purposes. So yes, so a real concern with a lot of these commercial ancestry tests is what happens to the data after you've gotten your little map, right? Because they still have it. Um, but a range of responses there, which again is not surprising, because people do have a range of perspectives on this. Um, you can, I think, learn some really interesting and positive things about yourself. Someone referenced like reconnecting maybe with a heritage that they didn't feel strongly with, and finding relatives they didn't know about. On the downside, sometimes these tests, and there's a lot of articles about this you can read in the popular media. Sometimes people learn things they were not prepared to learn. Maybe their parent is not who they thought they were. Maybe their relation to other members of their family is not who they thought they were. Maybe they discover they have a half sibling or a full sibling they didn't know about, which, is not uncommon. So sometimes the things that are uncovered in these tests are not at all what people expect, and that can be quite challenging. Um, so range of perspectives, as with all of these intersections with personal genetics. So the process, just very in brief, for anyone who is not familiar with it, um, uh, it begins with providing a DNA sample to a testing company. A company zeroes in on tiny fractions of the DNA where variation is high among people, and those DNA variants are then compared to reference populations, which are populations that can demonstrate, or there's documentation that they lived in one region of the world for several generations, and that determines which populations a person is going to be statistically associated with. And although hundreds of thousands of DNA sites on a particular genome are analyzed, these tests are only providing probabilities of ancestry, not definitive percentage. So critically, in thinking about interpreting these tests, I'm going to show you an example in a minute. Um, these reference populations are constructed using DNA from modern day people, not from those who were alive during ancestral times. And migration is a common theme throughout human history. Social identities change over time. Italian is not a biological category, but that's a category that might get used in ancestry testing, right? So because these different companies you have different reference populations and different kind of protocols for how they connect individuals to particular reference populations, a person's ancestry testing results can vary depending on which company you use, and sometimes with the same company over time if their reference population gets updated. So there's a lot of complexity in this, and I don't want to suggest um, that it's not real, but it really has to be contextualized in understanding that like this is a process and it's like statistical associations, not a biological truth that's being presented to you. Um, and as reference, like this can sometimes lead people to learn exciting information about biological relatives. Also, sometimes very unwelcome surprises, things that may be surprising or disturbing elements of their personal or family identities. And it's also true that for some communities, these tests or particularly communities that have kind of a fraught relationship, marginalized communities that have kind of a fraught relationship with science because of historical and ongoing issues, in particular, indigenous communities. A lot of these tests are not viewed favorably because there is a lot of concern that these tests are basically trying to resurrect this idea that of like blood quantum and that for many particularly indigenous, but not exclusively indigenous communities. Cultural connection and recent familial lineage are much, much more important in identifying, what it would mean to be, for example, Native American or, to be a member of a particular tribe. That regardless of what your DNA says, it's much more, you don't get to claim that you are indigenous unless there is an indigenous community that claims you. Um, and so that kind of layer is also very real, um, but is often part of people's ambivalence or uncertainty about how much meaning we want to ascribe to these tests. As just one example, so this is an actual result for a former member of the PGED team who had their DNA submitted to two different testing companies. So these are two completely distinct um, companies, completely distinct reference populations, and these are their results. And I'll pause for just a moment and encourage anybody to share. Does anything jump out at you about these two ancestry maps? Similarities, differences? Because there is some overlap. Well, mindful of time, I'm gonna keep going here, but feel free to note in chat. So basically, one is framed much more about current national boundaries, and one is takes a much more regional approach. Um, there is some overlap in sort of the parts of the world that these two tests connect this person to, but it is definitely not complete overlap, and it's definitely not complete alignment in terms of which parts of the world and their probabilities that are assigned to different parts of the world. And that again is just a reflection of the fact that these are comparing a particular genome to particular reference populations. Um, it is interpretive. It is not some biological truth that is being revealed here. And it just also really highlights the very strong element of migration and interconnectivity that many people around the world will their genomes will reflect. Okay, and then very briefly in our final minutes before we switch to Q&A, we'll be looking at the role of personal genetics in privacy and law enforcement. I do want to provide a content warning that I will be making reference to a real-life violent assault case, although nothing explicit, just the existence of that that case. As a final poll point, consider this question, agree or disagree. Law enforcement should be able to search private companies' DNA databases to identify potential suspects and victims of violent crime. So law enforcement should be able to search private companies' DNA databases, so private information in private hands to identify potential suspects and victims of violent crime. Okay, a few more seconds here for people to respond. Okay, so those are the results there. So again, range of perspectives. Someone notes only an extremely well-regulated context, and so if unfortunately, effectively no, because regulation is this is a challenging environment for regulation. Um, Right. So this is a pretty fraught thing, right? How do we want to see this incredibly powerful tool used to address societal problems and challenges? I want to note, just for purposes of disclosure, my partner works in a state crime lab, so um, the types of applications of genetics in law enforcement is something that I hear about a lot on a pretty close pretty close remove. Um, so, learning about your DNA is highly personal. The ability to do so raises complicated questions about how we want to handle genetic information. Who should have access to your DNA? What privacy concerns or restrictions should be in place? In law enforcement contexts, DNA is routinely used to identify suspects, find missing persons, and also to exonerate individuals who may be wrongly convicted or accused of a crime. And some people go so far as to say, believing that having a database of, all citizens or, all communities' DNA could be a powerful tool for public safety. Others raise very serious concerns about how such a database could be misused to have, a data bank that has biological information about, all people in the state of Maryland or all people in the United States. How could something like that be subject to abuse? And as we've seen in this presentation, scientific advances of the last few decades have really brought us to a time in which individuals have more access to their genetic information than ever before. They can have access to their relatives' genetic information, as we all share a portion of our DNA with our biological relatives. And a consequence of that is that analyzing our own genes or putting our own genes, our own genetic information into a database, also provides a lot of insight and an inroads into the genes of relatives and vice versa. So because of this familial connection, law enforcement officers have been able to identify suspects of crimes or victims of crimes by using information from their family members' DNA through a technique called familial searching, even if the person in question has not submitted their own information directly. And this method requires investigators, or one approach here, is to upload the DNA of a suspect or a victim to an ancestry or genealogy database. Then they create a fake profile for that person. And by using familial connections generated by the service, they can create a family tree of information. So if you have someone you don't know and you put them into a database, you can figure out the identities of some real people who must be related to this person you don't know. So that becomes a very powerful tool to figure out who that person you don't know is. Because you can figure out, well, we don't know this person, but we know their brother was, this person, or their great uncle was this person. And that provides a way to figure out who is that unknown person that we have a sample of. This was the process in the Golden Gate Killer case from a few years ago. The biological information from the unknown suspect or the ultimately convicted murderer to an open source genealogy database called GED Match. Um, they found a genetic connection in the database, a distant cousin of the suspected murderer, and using genealogical research, they were able to construct a family tree, narrow down the possible suspects, and finally make an arrest. This database was not developed to be a law enforcement tool. Um, in the months after the arrest of the suspected Golden Gate Killer, law enforcement agencies used the database to make arrests in several other cold cases. And many people feel that, this is a method that's justified in their pursuit of solving serious crimes. Others raise, concerns about the fact that, decisions about this was really in the hands of private individuals. And someone else noted, we may agree or we don't agree, but actually we should accept that it should be something we agree about. Yeah, that it's a really challenging place because, you know, I don't think anybody is, pro pro violent assault and murder, right? And yet we have to balance those, or arguably as a society, like, we have to balance those concerns against the very real potential for abuse. I can share that I've given this presentation or a version of this presentation a few years ago, and someone disclosed in the conversation that like they had a family member who was a victim of violent crime and lost their life, and their family suffered for years not knowing who not seeing justice for the loss of their family member. And tools like this are incredibly important to them in providing some closure. And that's very real. People's perspectives on that are very real, as are the very real concerns about how could something like this be harmful to individuals and to communities. And it's not easy answers. So with that, and that we're a few minutes over what I aimed for, but that's okay. Whether we're seeking answers to medical questions, tracing ancestry, or thinking about broader issues related to privacy, Personal genetics is intersecting and playing an increasingly significant role in all of our lives, and we should be talking about it. And our program is really seeking to find ways to support that in all the places that we can. As we wrap up my presentation, I just encourage you to think about other ways in which genetics is impacting your life now or might in the future. Think about things that surprised you in what we've been talking about today, maybe topics that you'd like to learn more about that our time together today has sparked. And thank you for coming. I do strongly encourage you to check out um, our website, which again, everything I've talked about has lesson plans, fairly up-to-date lesson plans attached to it. We have a card game for classroom activities. We have graphics. We are happy to do classroom visits, anything we can do to kind of help broaden conversations about genetics wherever we can. I'll also put my email in chat here as we wrap up. And with that, we have at least a few minutes for Q&A. And thank you, Sarah.
Awesome. Thank you so much. This was really cool. If anybody has questions, you can put that in the Q&A or the chat, ideally the Q&A so I can see it easier um, at this time. Any questions?
Oh, okay.
Here we go. Leah's working on it. Okay. And Sophie might be as well. This is the difference between having a Q&A with adults and with third graders. The third graders, it's just like I get like it's like 200 questions. What's the biggest genetics? What's the smallest genetics, etc. Okay. Oh, Michael Richardson's students have a question. Um, Michael's students wants to know why do we all look different?
Right. So, what is really interesting about humans is that we have like we do look different. As you look around the world, like there are people around the world, people look different. You can have like, we've all had the experience of like, oh, those two people must be related, things like that. There's family resemblance. People from different parts of the world may have similar hair color or, hair texture or skin color or facial features. All of that is very real. And in some cases, we can link that to particular genetic variants. We can talk about, genetic variants. I have a friend, Tina Lasisi, who studies the genetics of hair color and skin color. And this is something where, like, is of active research among scientists. Why does hair grow differently for different people in different parts of the world? And what's kind of can we find like an evolutionary story behind that? Um, and so a lot of those differences are real and have, some genetic or biological basis. And it is also worth remembering that humans are a very, very recent species on Earth, that when you compare the genetics of humans and, for example, chimpanzees or gorillas or other apes, our closest living relatives, humans are not very genetically diverse, that there's much more genetic diversity between chimpanzees in Tanzania and Uganda or uh, Tanzania and Senegal than there are among all the humans on Earth. And that's because we're this very recent species that exploded across the planet and occupied all these different parts of the world. So we do have real biological differences. Some of those differences can be explained through genetics, but it's also true that the story of our genetics is that we are all so similar, much more similar than most species on Earth are, because we have a very recent origin in the story of life on Earth. And I'm happy to talk more about that, but that's its own presentation.
Awesome. Thanks.
Um,
We got a
there was
question.
another comment about food and genetically modified foods. Yeah, like genetically modified foods, I think, are becoming a bigger topic. I think many people, and this is not going to be a surprise, like many people really find a lot of excitement in the idea that like we could create crops to become more nutritious or more drought resistant, things like that. And gene editing allows like interventions that are can be pretty dramatic and impressive. Um, and there can be some potential risks. You are modifying natural systems in ways that would be hard to undo if there was a concern that you uncovered later. And as a safety issue, genetically modified foods, in my view, there's usually not safety issues per se, but there is a potential for like introducing allergens, depending on what kind of modification happens. That is a real cause for potential concern. And so that's the kind of thing that like people should be informed and be talking about as politicians and others set policy. But that it is definitely related to what we're talking about today, and it is definitely something that people should be talking about.
Awesome. Thank you. We got a question from Leah. What kinds of understandings do you see in people about the relationship between DNA and other aspects of their biology, like proteins?
Um, what aspects? I think that something that a lot of people who work in the genetics field or who teach genetics recognize is that we absolutely do need to be talking about genetics. It's a very critical, central element of biology. And there are some real issues with how we tend to present genetics that gives people um, a flawed or incomplete understanding of how it works. I referenced Punnett squares earlier at the very beginning. Like people think of like Punnett squares. Those very simple Mendelian rules are real and important to understand genetic systems. And it's also true that those genetic systems are not typical of how genetics works in living systems. It is usually not the case that you have these very clear one gene, one outcome relationships where you can, put it into a single Punnett square and say, okay, it's, three quarters this phenotype and one quarter this phenotype. And there's a real recognition, a growing recognition among educators, science educators, and scientists that we need to find better ways to teach that because DNA is real and important and obviously has an impact on our biology. We have to understand it. But if the Punnett square and Mendelian genetics is all you know about genetics, that's a very limited and maybe harmful um, framework for thinking about human diversity and
Yeah.
complex traits like health and intelligence and physical capability or ethnicity. And that kind of answers your question. Like DNA is about ultimately like genes are about generating proteins. Like right, that's the product. It's one step of this very convoluted thing. It's one element of that. But I think DNA tends to be reified or held it to like as a more magical element of the system, but it is just part of this much bigger system that is hard to summarize for 10th grade science in a way that is true but contextualized. Yeah.
Yeah, it seems like just the Mendelian version is what gets into like the American Eagle ad, right? It's like
Right, right, right.
you don't get the full complexity. Our ASL person had a hard out at one.
Yeah.
So unfortunately there's no more ASL interpretation. Sorry about that. Yeah. Okay, we have oh, we do have more questions. Um Ooh, from Mandy. When they edited the bone marrow for sickle cell, did they have to go in and edit each cell?
So my understanding, I hope I can get the details right. My understanding is the treatment process is you basically wipe out through radiation or chemotherapy existing bone marrow or as much as possible of the blood producing bone marrow cells in some of the long bones, which is where most of these are. You don't necessarily have to get every single bone, every little nook and cranny, but you got to get, the big bones that have lots of bone marrow, lots of blood, lots of red blood cell generating cells. And then you replace those with gene edited cells that are going to be generating red blood cells that do not have the sickle trait. So by doing so, you greatly reducing, if not nearly completely eliminating, the sickled cells that are circulating in the body as the body as the new gene edited cells kind of establish themselves in the bone marrow, that then kind of becomes what the body produces. So you're not editing what's called germline. So this is editing a person. You are not editing their descendants. You're not editing. So the treatment for sickle cell in this case is um, only that specific person. It's not something that can be inherited. Um, and um, so that's the process, as I understand it. And there's other approaches to gene editing, but in that case, that's the process, as I understand it.
Awesome. Let's ask one final question and then call it for time, because I think this is a good one. Anne Sophie asks, what's the biggest misunderstanding or disinformation that you've seen circulating about genetics recently, and how can we approach it with our students?
I think it's what I mentioned just a moment ago. I think the idea that Mendelian genetics are real, but that they are not how most genetics work.
Right.
And so the challenge for educators, I think, is how do we teach this in a way? Because the Mendelian thing is important and helpful in understanding how genetics works.
Mhm.
And if that's all you know, if that's what you take away from how genetics works, that's actually
It's
quite
basically
harmful because
wrong.
it's
Yeah.
incomplete.
Right.
It's woefully incomplete. So I think I would to pithily say it's that Mendelian genetics are real, but they are not how most genetic systems work.
Right.
And
Great.
there are resources, and this is something our program has explored. There's a team at Duke University, a colleague of mine, Elaine Guevara, and colleagues has developed some lesson plans to kind of explore that. So if you search for like Elaine Guevara, Duke genetics education, something like that, you can find some of their free online resources. There's another researcher, Donovan, who has worked on this exact issue about kind of how do we teach genetics in ways that don't leave people with very harmful and incomplete understandings of genetics. So these resources are out there to kind of help educators find their way through it. It is hard because the impulse, of course, is always to teach the simple and work to the more complex. But you can't just stop at the simple, unfortunately, or that becomes really incomplete. So there are resources out there. Our program has some. Other programs have some. So I would just look for ways to incorporate that understanding into your pedagogy would be really critical.
Yeah, especially for high school classes, that might be the last science class your students
Exactly.
ever have. And so if you leave it at
Exactly.
the incomplete point, it's a problem. It's a problem that then I deal with on the street 15 years down the road when I'm talking to people who haven't done science in 20 years. Um, awesome. This was really cool. Thank you for sharing all these resources. We will, for everybody here, if you'd like to share this with one of your colleagues, it'll be posted to the Skype-A-Scientist YouTube in sometime this afternoon. Um, and we'll include resources in the YouTube description as well. So if you want to tag some stuff, get that. That sounds good. All right. Well, thank you again. This was really
Thank you.
awesome. I'll see you soon, I hope. Bye.
Bye-bye.